Hello, thank you for joining us for our webinar. This month, we are pleased to have Dr. Susan DeLong with Colorado State University. Susan is an assistant professor in the Department of Civil and Environmental Engineering, and she earned her PhD in engineering from the University of Texas in Austin. During the talk today, we have a really interesting discussion that Susan's going to do about metagenomic tools that we can use for research to help us elucidate key genes that are important for qPCR analysis. So we know in the industry that qPCR is the gold standard that we can use for quantifying different microorganisms and different genes of interest. But in order to design these assays optimally, researchers have really state-of-the-art metagenomic tools that we can use to aid us in this biomarker discovery and assay design. So Susan's going to talk a lot about this for us today. And this is going to show us how we can do an even better job in providing a comprehensive assessment of contaminant biodegradation in the environment. So before I turn it over to Susan, as many of you know who have attended before, the webinar today is being recorded and it'll be available on the resource section of the Microbial Insights website. We'll also send out a link to all registrants as soon as it's available. And at the end of this webinar, we're going to have a question and answer session. So if you have questions that you think of while Susan's talking, go ahead and enter them into the question section in the GoToWebinar software. And at the end of the presentation, Susan will go ahead and answer those. Also, if you have additional questions that you think of after the webinar, feel free to email Susan or info@microbe.com and we'll get with Susan and we'll get those answers for you. So without any further delay, I'll go ahead and turn it over to you, Susan.
All right, thanks, Dora, so much, and thanks everybody for joining today. I'm excited to tell you about some of the opportunities available to us using meta-omics tools and some of the data that we've gathered so far. So today I thought I'd start by just reviewing a little bit about molecular biology tools and the different types of assays that we can use to get all of our brains on the same page. I know everyone's coming from a little bit different perspectives, so I thought I'd begin there. I'm going to talk then about assay accuracy and some ongoing challenges and, challenges and opportunity for improvement. So I'm going to talk about how we're using some of these new tools to develop new assays and better assays, which is really exciting. And then our specific topic for today is really meta-omics and how this new field is allowing us to design and develop new assays, and then we'll have questions at the end. So we all know the issue. We're applying microbial tools that target biomarkers to develop lines of evidence for bioremediation at field sites. And there's really kind of two general types of tools that we can track. We can track specific organisms at the level of phylogeny or taxonomy. We're just identifying the types of organisms there. Or we can target a function or process by using qPCR assays that target functional genes. And we're mostly focusing today on the latter because those types of assays are more informative generally. All right, so just to remind everybody a little molecular biology, DNA has, the genome contains all of the genes that are the potential for an organism. When those genes are being used or expressed, you get RNA transcripts, and those transcripts are then translated into proteins, which are the enzymes that catalyze reactions. So a lot of the work that we're going to do today, or that I'm going to talk about, is at the level of sequencing DNA, but then to validate that these genes are relevant and active in the field, we're going to want to look at RNA. And ideally, we could also look at proteins, but so far we're mostly looking at RNA to validate these. So to summarize, the proteins are really catalyzing the reaction. So we don't want to just look at DNA. Ideally, to validate these new assays we're developing, we want to really, because we're discovering them with a new tool, we want to be able to verify that what we've discovered is truly relevant for these degradation processes. So again, we can look at these, we can use these biomarkers that target fingerprinting biomarkers like the 16S ribosomal RNA gene. That's really just telling us who is there. If we want to know what they can do or what they are doing, we're going to want to target these functional genes. So the current challenges, and I mean, it's really exciting to me what we've been able to do with molecular tools and the availability of assays already. But from the research perspective, I think of it as, you know, a menu of assays that you can order from to analyze your field sites. And when we think about the microbial potential that exists on this planet, I would say that our menu is still limited. There's a lot of new microbial diversity to be studied and characterized. And so I think that in the coming years, we're going to see more and more new assays that are just really going to make these tools even more useful for practitioners. And then the other issue that I want to talk about today is assay accuracy and the possibility of false negatives, because I think the meta-omics tools are also really helping us expand the range of organisms that we can detect so that we will have less risk of false negatives. So I'm going to talk through why I think that's the case. All right, so just to think about the background here, molecular tools have historically been based on pure cultures, or at least to get started, researchers have worked with single strains that they were able to grow in the lab, and they've done a lot of time-consuming development of to determine sequences that we can use to develop qPCR assays. And if you base an assay on a pure culture, there's a risk that that assay will be inaccurate when you go and apply it to the field. Now, we've done more and more sequencing of mixed cultures as the years have gone by, and then people will iteratively improve those assays. And I know Microbial Insights does a lot of work to improve assays that are published in the literature to make sure that they are field relevant. But at the same time, we find ourselves in this sort of catch-22 where we can only develop assays based on sequences that we have. And so the more sequences we get that are field relevant, the more we can improve those assays. So so often what researchers will do is get more and more pure cultures, get all of those sequences together, design assays that target a lot of different strains, and either they can define one assay that targets all those strains, or they have to run several different assays and add the numbers together. But ideally, we could go after field relevant mixed microbial communities, so communities that have many different species in them from the get-go, and get at the sequences that we want to target in these to develop these qPCR assays. So this is just a figure, for example, that just sort of highlights the diversity of organisms that might be involved in, just as an example, anaerobic hydrocarbon degradation. And this comes from a paper from Rabas et al. And you can see how the primers, they have to design a series of different primers to target this diversity. And again, you can develop those primers when you know what the diversity is and what the sequences are that you're trying to target. So let me just tell you a little story about the accuracy of qPCR assays. This data is from a paper out of my group. Brett Ledeker was the first author on it, and it was focused on a target for perchlorate reduction. So it's a specific case, but I think there's the possibility that this story repeats itself for other contaminants. So on the vertical axis, you see quantification accuracy, and on the horizontal axis, I have a series of synthetic microbial communities, meaning that I went into my freezer, took out 4 different strains that were known to reduce perchlorate, mixed them in different ratios, and I just kept it simple here, not to get into the details of which species were more abundant in which sample. I'm glad to send you the paper if you're interested in this particular story. But what you see is depending on the composition of the microbial community, we get different accuracy for the assays. So and the reason for this is that the sequences of the target genes in the different species vary a little bit, enough that the primers, which have to bind to the target due to their similarity, their complementarity, have mismatches. So bases that should match, and A should match to a T, they're not matching. So if you have an A on the primer, you have a G where you're trying to bind, and so it's not binding properly. And these mismatches we found can reduce accuracy 2 to 3 orders of magnitude. And sometimes we can get these reductions in assay accuracy even when those mismatches are in regions of the primer, such as the 5' end, which have historically thought to be not that big of a problem. We can also see that sometimes we got a non-detect with this minimal number of mismatches. So this can happen if we have a target gene where it's highly variable, the sequences are highly variable in the field, we might sometimes get a non-detect because our assays were designed on slightly different organisms that have been cultured either in pure culture or a mixed culture in the laboratory. And so we don't know that we have a false negative. And so this, of course, is a problem if you're trying to develop a line of evidence for degradation at your field site. If your organisms are a little different, a negative, you might get a negative even though you have that, those degrading organisms there. So I always tell people a positive is pretty much a positive, but a negative, might actually not be a negative because there is this possibility that your organisms are just a little bit different. So again, the more we learn about these organisms in the environment at field sites, the more we can expand this database and redesign primers to address this issue and have less and less false negatives. And so that's what we're trying to do. But we're trying to do it in a really efficient way. So in comes omics. So we all, you've probably run across this term omics, and it really encompasses 3 different types of omics tools. So there's metagenomics, or genomics or metagenomics. So metagenomics simply means, the meta part simply means that we're working with a mixed community of organisms. So genomics sequences all the DNA in one organism. Metagenomics will sequence all the DNA in an entire microbial community. People have often used this term to talk about sequencing all of the fingerprint or 16S ribosomal RNA genes in a community. But really, at its roots, metagenomics means sequencing the entire genomes of all the organisms that are present in a site. Then transcriptomics is sequencing all the RNA of one organism, and metatranscriptomics is sequencing the whole community. And then proteomics is detecting all of the proteins in an organism, or you can do that for an entire community. So this is the omics set of omics tools. What I'm going to talk about today is mostly using genomics to develop databases to improve tools, but we're also doing some work right now with transcriptomics, and on some tangential projects we've been using proteomics. And all of these are relevant and going to be at play for the remediation field, I think, but especially genomics and transcriptomics are a little easier to apply. Okay, so we can use targeted assays, which are really just getting at one part of an organism, or we can sequence, and we're not doing anything targeted, so we're able to detect everything that's in that community without having to know what we're looking for. So I just always show this slide, and the sequencing costs are have gone way down. And so a lot of this sequencing technology is really available to the remediation community because it's affordable now, which is exciting. All right, so I wanted to talk a little bit about the different opportunities that we have with using next generation sequencing, and then I'll get into the specific application that we are working with. So people are often using sequencing now to sequence the 16S ribosomal RNA gene, which gives us a picture of all of the microbes, and I put all in quotes because nothing is perfect, and there's a lot of low abundance microbes that you might not end up paying attention to. But theoretically, we can sequence all of the species that are present in the sample. And so instead of just having quantities of a specific target species, which you would get from a targeted assay, you're going to get this whole picture of the microbes present. And again, sometimes people refer to this as metagenomics, but it's right now what I'm talking about is just sequencing that one marker. So we end up with the names of the organisms that are present at a site. So then we can try to use that data to build site conceptual models or conceptual site models. And then the question arises, well, is the phylogeny or the type of organism you detected really predictive of the processes you use in the field? And sometimes the answer is yes, and sometimes the answer is no, that phylogeny and function are not coupled in that way. So it's a useful tool. I do it myself. This data is from one of my papers, but there's an opportunity to get better data. So if we want to go back and target those functional genes, again, we can have targeted assays that track a specific functional gene, or we can do metagenomic sequencing and sequence all of the genes in all of the genomes of all of the organisms present at the site. And we can map that, a first cut at analyzing the data is to map that to these KEGG pathways. And you'll see some data there on the right, which is all of the pathways that were detected in one of our organisms. So you get it all. You don't have to order off of a menu. Of course, there's some caveats here. First of all, you can look at that data and like, wow, what do I do with that? There's a lot there. You can zoom in to specific sections of the map. So the lower left-hand corner of the figure that I just showed you, if you zoom in on that, you'll see that the pathways are pathways for xenobiotic degradation. And there are some, contaminants that we all recognize here. And if we detect those, so in this example, there'll be a red line over a pathway that was detected in our data set. This is just an example. This is an annotated database that we're mapping to. So we're taking our data and we're querying it against knowns. So fundamentally, we're limited by what's known. So if we don't know what the genes are for the degradation of a particular contaminant, or there's new pathways that are undiscovered, we will not be able to say anything about that based on metagenomic data because there's nothing to match to in the database. So you can see the names of contaminants and pathways that show up in this xenobiotic degradation and metabolism section, and it's certainly not everything that we deal with in our community, but there's some good stuff here. So then the idea might be that we could go to field sites and get samples with depth or different zones or time or whatever you want to do, and you could sequence the entire metagenomes and you could map it to these pathways and you could look at what genes are expressed. But the issue is, realistically right now, that's pretty expensive, not only because the sequencing of all of the metagenomes is expensive, there's a lot of bioinformatic time in processing the data and understanding what it means. So we're thinking about this from a slightly, we're thinking about how we can use these tools in a practical way. So if we apply targeted assays and that focus on a process, it's pretty affordable. I mean, you know, you can send your samples out and get nice data back for a pretty reasonable price. But the catch is that we miss anything that we're not looking for directly or that we didn't even know about. So how can we use these metagenomics to address that problem and improve these tools over time iteratively? What we can do here is apply the metagenomics to find new field relevant sequences and then circle back and develop new targeted assays. So we improve the menu of targeted assays, and then you can apply those to as many samples as you like cost effectively. So we don't need, you know, in my perspective, we don't need to go and do metagenomic sequencing on every sample at every site. We need to identify representative samples or communities from sites where existing assays seem to have a problem, like we're getting the data back and it doesn't seem to make sense with other parameters that we're monitoring at the site. And we can sequence the metagenome of one or two of those samples, figure out if there's a reason that the assays are giving us data that doesn't quite add up, and then circle back and improve the targeted assay. And then we can apply that targeted assay, you know, to as many samples as we want at as many field sites as we want. So I think that's really one of the exciting directions that we can go with meta-omics tools in our field. So how does this work? So we've, you know, go to a field site. We've been working with hydrocarbon degrading microbes. So there's a L apple there. We have a hydrocarbon degrading mixed microbial population. We're going to isolate DNA from that culture, run it through a next generation sequencing protocol, and then we're going to use bioinformatics to find new targets and sequences, which is, you know, a complex process. So we have all these collective genomes. We're going to fragment it. We're going to sequence it. And then we can query those sequences against the database to identify genes. But really, this is not an automated process where you just get your answer and you can go apply it to site decision making. You really need expert analysis and an evaluation of the annotation in the databases. Sometimes the genes you need are actually hiding in a place that's different from what you might have expected. And then we really need to test the hypotheses that are generated out of the metagenomic sequencing before we go back and apply these assays at field sites. So what we have been doing is working with a model system that was shared with us by Elizabeth Edwards. Thank you so much to her. This is a field-derived methanogenic culture that degrades toluene and O-xylene. And interestingly, after all of these years, the O-xylene degradation pathway for this organism was not fully known. It had been hypothesized that it was analogous to the toluene degradation pathway, but as I'm going to tell you, it's actually just a little bit different, such that we would have to modify assays. So even though this culture has been around a long time, there were still some interesting things that we could learn from it by applying these meta-omics tools. So this is the 16S data for the culture. So this figure shows the relative abundance of bacteria and archaea in this culture by sequencing that biomarker. And it's a methanogenic culture, so we have both bacteria and methanogens in here. And this is helpful because we can understand, you know, how similar or different this culture is from other field sites, that you really can't, you know, from this data infer details about the process, but it's the context of our system. So we sequence the metagenome of this culture, and here's all the metabolic processes that we observe. So this is our functional potential for the system that we observed. There's a lot there, right? So we need a strategic way to look at this data. We need hypotheses to guide our efforts so that we can extract something useful from all of those red lines. So one of the first things that we can do is map that data to these KEGG pathway categories. So the annotated databases have been broken into these specific categories. So we can go in and see how many of our sequences map to these specific known functions like xylene degradation, toluene degradation, etc. And Karen Rossmessler, who worked on this project and did most of the work really, went in and manually annotated this data to distinguish between anaerobic and aerobic genes, so we can see that. But interestingly, when you look at this, so we're working with a xylene degrading culture, an O-xylene degrading culture that's been enriched for a long time. It's pretty specialized, for that process. And when you look at the amount of genes that map to xylene degradation based on this default annotation, there's really not that much there, which means that the important genes could be hiding somewhere else. And that's what we found. They're probably hiding in the toluene degradation category. So we really have to dig in with some more, targeted approaches to extract what we need. We can't just run this through existing algorithms and there you go, publish a paper. So we had a hypothesis for this system. We expected that the anaerobic toluene degradation, the anaerobic O-xylene degradation was mediated, that the first step was mediated by a fumarate adding enzyme, benzyl succinate synthase. And so I'm showing you the pathway for toluene here. And because we've hypothesized that the pathway is similar, we think that that first gene in the pathway will be similar and that it will be a benzyl succinate synthase. So what we did is we specifically went into our data and pulled out sequences that looked like benzyl succinate synthase. That's hard to say fast. Benzyl succinate synthase, BSSA, and then we spent some time looking at those in detail. So what we found was 4 different, what I'm going to call a contag. So when you do your metagenome sequencing, you get fragments of sequences, and then you have to stitch that puzzle back together. And the pieces that you get, you don't get the whole puzzle stitched together. You get pieces of the puzzle, and those pieces are called contags. So those are, sequences that are of enough length that you can pretty confidently identify, something about their function. So we found 4 genes that look like benzyl succinate synthases or fumarate adding enzymes. And there are a series of different types of genes that encode fumarate adding enzymes for anaerobic hydrocarbon degradation, depending on what the target molecule, the target substrate or contaminant is. So BSSAs have been linked with toluene degradation. There are NMSAs, which have been linked with naphthalene degradation, and ASSAs have been linked with alkane degradation. So in this tree, where we're looking at the similarity between our sequences and known sequences, you can see that we, in our culture, identified sequences that fit in these 3 different groups. Interestingly, though, the contag that mapped to, that was similar to an NMSA, was by far the most abundant in this culture that had been highly enriched on O-xylene. So we thought, maybe this contag, even though it, is annotated as being similar to a naphthalene fumarate adding enzyme, perhaps this is actually involved in the O-xylene pathway. And the other enzymes that are, the other genes and their associated enzymes could also be involved, and we wanted to look at that. So there's existing assays to target fumarate adding enzymes, BSSA genes, and this category of enzymes is already known to be relatively diverse, and it takes a few assays to actually encapsulate that diversity. But even then, we found in this particular culture that none of the existing assays would work well. We don't know how common, we're still exploring how common this particular type of gene is in the environment, but from our culture, we map it to known genes and we find there was a lot of mismatches. So this shows you 4 published qPCR assays and then compared to the 4 different contigs we found, and then in the middle you see the number of mismatches with the forward primer, comma, the number of mismatches with the reverse primer, and anything over 3 is not good. So these existing assays just wouldn't work, which is fun and exciting if you do research for a living, and not so convenient if you develop assays. But the important thing is that we can use this new information to improve these assays and develop new assays and expand beyond what's was already published in the literature. So our approach now, so we've discovered these new contigs, but we really haven't shown that they're relevant to O-xylene degradation, except for the fact that they were highly enriched and abundant in our culture, which had been growing on O-xylene for a long time. But still, we pulled these out just based on the hypothesis. So we want to show that these new genes that we found were actually expressed when O-xylene is being degraded. So we grew cultures on glucose as a comparator, and then O-xylene, we extracted RNA for expressed genes, and then we did RT-qPCR and compared expression. So the question was, are these genes specifically expressed when O-xylene is degraded? So we tracked degradation of the substrates. So we looked at, we made sure that our cultures were actively degrading glucose at the time that RNA was extracted. We made sure that our cultures were actively degrading O-xylene or toluene when RNA was extracted, and so we have this data here. And then we quantified expression of these new genes that we found, which are M122 in blue. There is the one that is similar to an NMSA. And then M104 is similar to the BSSA, which is what was originally hypothesized to be involved in O-xylene degradation. And then M107 is the ASSA. We also used the currently available assays, even though based on our in silico analysis, we didn't expect any of those assays to work. We figured, what the heck, we'll run a couple of them. The only one that worked was this SRB that I'm showing in yellow there. We did get a detect. I'll show you this on a log scale in a moment. But you can see that NMSA, the M122 gene, was highly expressed when O-xylene was being degraded, and it's also differentially expressed, meaning that it's much more highly expressed, much more abundant when O-xylene is degraded relative to toluene or glucose. So based on this data, we think that in this culture, it is encoding the enzyme involved in the first step in O-xylene degradation, and therefore likely to be useful for the field. So it's 10x more abundant when O-xylene is degraded and 3.5 times more abundant than when toluene is. It's 3.5. It's actually differentially expressed when toluene is being degraded as well. So compared to glucose, so it might be involved in toluene degradation, or they might just be co-regulated. So if we look at this on a log scale, you can see. Well, here, let me go back. So you can see that the BSSA is also highly, well, it's much less expressed, but it is readily detectable and more expressed when O-xylene is degraded, but even more expressed when toluene is being degraded. So perhaps that's more relevant to toluene degradation, which this culture has been grown on over the years. And then M107 is also detected. Perhaps, again, it could just be co-regulated, or it might be involved and it might be present in lower abundance members of the community. The BSSA data from the known assay could, my guess is that those numbers are not accurate, and we're actually detecting something else. We're detecting, say, M104, but just with less accuracy. So that just shows you, what you would get with available assays. All right, so I wanted to tell you about another tool that we're using to validate these new genes and their role in degradation in the field. So we've been doing some work with computational enzyme structure modeling, and I've been collaborating with a colleague of mine here, Chris Snow. And what we do is we take the sequences for our new enzymes and we predict protein sequences, and then we model the structure of the protein using existing so laboratory validated protein structures. So other similar proteins that have actually been crystallized in the lab and their structures have been solved. We use those solved structures to constrain our in silico prediction. And we predict what the active site of these newly discovered enzymes looks like, and we model it with the substrates in there. So this is toluene and fumarate in one of our enzymes. And then we can use these models to predict whether or not the substrates fit. So I think this is another exciting tool when we're going out to the field, we're sequencing, we're getting mixed cultures, we're sequencing them with meta-omics. If we want to move quickly to get these assays to the field, excuse me, and trust that what we're monitoring is really relevant, it's great to have these computational approaches to validate it without 3 more years worth of lab work. So this is something else that we're working with. So meta-omics allowed us to detect the total metabolic potential of our community. It allowed us to find these new fumarate-adding enzymes, and it's informing the design of new assays. And we're having fun working with Microbial Insights to try to determine the relevance of the particular sequences that we found, and then perhaps expand the available assays as that seems useful. This data is going to be in a publication hopefully sooner than later. So keep an eye out or shoot me an email and I'll certainly send the publication your way. And let's see. So, if I could summarize our opportunities and challenges in the omics space, sequencing 16S ribosomal RNA genes with what some, are calling meta-omics is now really affordable. It can be applied to detect all the microbes that are there and the microbial potential, but you're really inferring the processes. Meta-omic sequencing is still pretty expensive, and really the computational time and the analysis that goes into it is probably more of a barrier than anything. But I really think that these tools are going to be useful when existing assays appear to fail or are not suitable, or as we move into some of the emerging contaminants and we're trying to design new assays, we can sort of from the get-go get at the diversity of the sequences that are in samples. So thank you. So a lot of the work that I talked about today was done by Dr. Karen Rossmessler. So postdoc in my group, I work a lot with Tom Sale for field samples here at CSU. As I mentioned, Chris Snow was working with us on the computational protein modeling. Big thank you to Elizabeth Edwards and Fei Luo for the culture. And then a few folks in my group who do a lot of great work, Maria Rani Reno, Diana Nunez, and Brett Ledeker. And then, of course, we can't do anything without funding. So thank you to all the folks who fund and support our research so we can make these advances. And I will take questions. I think I'm a little early, so we can do lots of questions, or you're more than welcome to email me, visit me in Colorado, and we can talk about how we can use all these exciting tools to make advancements in our field.
That's great. Thank you very much, Susan, for the webinar. It's very informative, and I think it's an interesting concept of, and you and I were talking about this a little bit before the webinar, is that, these tools are great. They give us a means to be able to truly, do some site assessment, and we're growing and changing continually on that. I guess, can you kind of summarize kind of your thoughts for the industry over the next 3 to 5 years, kind of what you see us doing and maybe going as far as some of more of the bioremediation assessment and truly being able to apply this to make it more actionable for what we're doing in site management?
Sure. I will try to take out my glass ball, but that's not too long of a timeline. So some ideas that I have of course, in the research space, we're interested in getting cultures from as many sites as possible, what funds permitting to do some of the sequencing and advancing tools from that perspective. I think also something that we could do, really partnered with industry and making this broadly applicable, as we identify specific contaminants for which these approaches would be most relevant and most useful, you could offer sort of a troubleshooting option where if the data comes back and doesn't seem to make sense with field assessments, you could choose specific samples, do metagenomic sequencing, query very targeted sequence groups like we did, which I think that we could develop pipelines to do that for subsets of targets. And then, it might be possible to have, that data from a single sample or do custom assays for sites where you really need to invest that additional money to get data that makes sense. So I think that, I think the workflow that we've developed could actually be adopted into a commercial space if we identify very specific contaminants where it's useful and build sort of automated pipelines to get us at least, right before assay development. And then if you're trying to develop assays just for one group of organisms and not trying to develop assays that target, 10 different organisms at once, that process is relatively simple. Of course, that's somebody's time, but I think that's a possible, type of workflow that we could see commercially. Of course, I'm glad to hear your perspective on that as well, Dora.
No, I definitely agree with you. And I think, like you mentioned, screening samples with metagenomics is a great way that we can make sure we're getting whatever, intended application that we have. So I have, kind of two follow-ups on this is, thinking about how to pick the right samples from our sites for our metagenomic analysis. Can you talk about some of the indicators that you would use? Like, would you look at geochemical conditions and select, some of the wells, or would it be based on location, like source area locations, or isotopic indicators? Like, what would you use as some of your screening criteria? And then when you finish that, I do have a second follow-up on that.
Sure. Okay. So first of all, let me just clarify that what we've done to date is using an enrichment culture. So we got the DNA out of a lab culture. You don't need that much DNA. So I think that we can imagine just getting DNA out of a field sample directly and sequencing it. The data might not look as pretty, and there might be caveats to that. But I do think we can sort of do what you're suggesting and pick specific field sites to sequence. Although, I just want to clarify that the data I showed was from a culture. Yeah, certainly. So, I mean, I guess I would choose the samples where you have other lines of evidence that the degradation processes are occurring, or where you have a solid hypothesis that such and such degradation process is occurring, but where existing assays maybe are giving you numbers that are lower than you expected, or you're getting non-detects. So that would be based on all the types of data that you just listed, right? Geochemical data, isotope data. Of course, and then what types of geochemical conditions are most important on a volumetric basis or for the contaminants you're looking at, so that the information you get is relevant to like the largest volume or the most kinetically important processes. Of course, if you have a really diverse site, if you could choose a representative sample from each of the sort of categories, redox categories, contaminant level categories, that would be best, because whatever conditions you have are going to have a selective pressure that will shape the microbial community such that you'll have perhaps different genes there. The other thing that might be useful is to do some phylogenetic characterization on a lot of different samples. So do the 16S sequencing so that you understand how diverse your microbial communities are, and then kind of bin them and choose representatives based on that type of data, which is much cheaper.
That sounds really good. Yeah, definitely. And as a follow-up on that, when you think about the different tools that are available, the full metagenomic testing from a commercial standpoint isn't readily available, looking at all the different genes. And then when you get that data, it can be overwhelming if you're not a practitioner that is doing this or a microbiologist. And that's one of the follow-up questions that we got is, how can people actually take this data and use it for something practical at the site, because they're not microbiologists, and the jargon that we use when we from a microbiology standpoint can be pretty overwhelming. So how do you transition that to something practical for regulatory agencies to take action or to approve, decisions based on that?
Yeah, well, I think that's the opportunity spaces for companies, like Microbial Insights, right? You need an expert. This is not, I can't do, I'm not a geologist, and so I work with people who are geologists. And I think you're always going to need that in this space. You're always going to need the expertise of a company or academic partner who runs this and does some interpretation of the data and processes it into a form that is digestible by people who have different expertise or by regulators. The easiest path I see is to take that data and then do a quantitative PCR assay, because we're used to that type of data. It's quantitative. We can compare it to thresholds or, expected levels of a target gene. Metagenomics by itself is not quantitative. Really, I mean, we have relative abundance of genes, but it's not telling us copies per gram of soil. So I don't think that it will be best to take metagenomic data straight up and use it for decision making. I think we want to take that information and then run quantitative assays, and then you'll end up with the type of data that we're used to. But I think that's going to have to be done by people with expertise. And we want that because if we automate all of our jobs and they're all done by robots, then no one will have anything to do.
And I think that's a good point, and that's why we work with researchers like you, Susan, is because as a lab, we need to identify these new organisms and genes that are of relative importance to the environmental application. And then with what we learn from you guys, then we can, optimize our assays to be even more comprehensive than they already are. So we're constantly evolving and growing what we can do as an industry, and that's one of the things I think you pointed out in the beginning, is that this is, changing very rapidly, and we're seeing a lot of growth, and it's a very exciting time. But I think really what we're producing from these qPCR assays, when we put it into context, is something that can be used as an action at a site. It's not data that doesn't mean anything. We do have an understanding of how to apply it to site management decisions.
Yeah.
Okay, so let's see. The next question here is, when we're talking about bacteria in general, this is kind of outside the scope, he's saying, of your presentation a little bit, but do you have any feeling or guidelines for the temperature dependency to get good active bacterial degradation for these contaminants in particular?
Well, somebody's been reading my papers or not, because I have a paper on that. So yeah, we actually did a temperature study, particularly of hydrocarbon degradation under conditions where there was L-napol present. And we found that the threshold, we looked at 4 degrees, 9 degrees, if I can remember correctly, 22 degrees, 30 degrees, and 35 degrees, as well as some higher temperatures, because we were thinking about thermal enhancement as an option, although the higher temperatures didn't work very well. And I can send anybody that paper who's interested. we found that the threshold was somewhere between 9 and 22, where we really saw the degradation kick on. And again, these were methanogenic. This was a particular site that was methanogenic. There was LNAPL present. We really saw it kick on. And if you think about it, 10 degrees or below and 4 degrees, that's like the temperature of your refrigerator. And ideally, microbes don't grow particularly well in the refrigerator. Of course, those are a different category of microbes, but it does kind of translate to the field. If we get it up to something like room temperature that we're comfortable in, we really see it kick on. And others have seen this as well. I've run across other literature in other types of systems where things really kick on at about 18 degrees. And then if you look across different geological, climatic regions, the answer varies a bit. So if you look at the Arctic, I forget what it is, but they kick on at like 10 Celsius, and then if you look at tropical regions, they can go even warmer. And if we kind of conceptualize this with a rule of thumb, we think that maybe about 5 degrees above Celsius above the sort of historical high of the site is optimal. Of course, that's just if you're trying to enhance the degradation. But the microbes have evolved over a long, long time to site conditions. So they're going to be happiest, not too far out of the range that they've seen in the past. If you get too hot, it's they feel like they're being cooked a little bit. And if it's too cold, then they kind of go into hibernation. So, ballparky 20 degrees is good.
That's great. Okay, I think this is a good follow-up to that is, when we've been talking a lot during the presentation about fermentation of hydrocarbons to methane. How close do you think we are to having some assays that can help us predict the process for natural source zone depletion? Do you think we're anywhere close on that?
Yeah, so you mean assays that work in the source zone, or is the question about rates?
I think it's closer to probably rates would be my guess of what they're asking here, and
sure,
trying.
That's everybody's favorite question.
Yeah, exactly.
Yeah, I know. Well, I think that over the years, we have seen that in some cases, the quantity of transcripts, so the quantity we would get out of an RT-qPCR assay, so targeting RNA, does correlate with rates. We have seen in other cases that those quantities are not correlated. So that's a fundamental basis for whether or not we're going to get there with these types, really put in the qPCR number, churn out a rate. If we don't have a correlation, if they're not fundamentally correlated, we'll never be able to have just one equation that spits out the rate. RNA is going to be a better predictor of processes than DNA. We do have methods to get RNA out of source zones, despite that NAPL that's there. So, we're certainly, I think, okay on that front. In terms of trying to understand when and where RNA transcripts are correlated with rates, I think there's more work to be done to understand when they are correlated and when they're not. So I think we could get there on that front. The other way I see it working is trying to have sort of a broader category. And I think you guys have seen this in D. helicocoides stories, where there's sort of a threshold quantity that means it's going to go. And then you can, expect the rates to be somewhere in the range of what people have seen in the past. And so it's a little, a number plus or minus in order of magnitude, rather than a really, number we can pin down and say, in 2021 we'll be done on February 7th, right?
Yeah, and we have seen that with the helicocoides, that there's a range, that we know this is kind of where you're going to get this abundance level of the organism is going to give us, a generally useful rate of degradation. From my perspective, I don't know that I've seen that with some of these anaerobic markers for BTEX degradation. Do you have a good feel for that from what you've seen?
Not really. I think it takes a whole lot more data at a lot more sites at a lot of different field conditions to start to see those trends, and also be sure that the data that we're trying to correlate is fully capturing the diversity of the site, so we know that the numbers we're trying to correlate, should be correlated because they're actually measuring what's there. I mean, I got into that whole story about the PCR assay because I had data from a bioreactor that was telling me that processes were going well, at least at certain time points, and then data I had from the published PCR assay was just nothing made sense. And it wasn't that there was no correlations or no patterns in the data. The assays were wrong, and so it's possible that as you refine it, the correlations will start to show up a little bit. But I think we have to have pretty big data sets. But I mean, you guys analyze many more field sites than we usually have. A small subset of model sites. So,
well, and we see that this is a relatively new marker, right? That we're just now understanding anaerobic BTEX degradation as a whole. Even though BSSA was identified a long time ago, we don't. Yeah. We just now understand benzene a little better, and I don't know that we know all the pathways for it yet. But we're starting to grow on that. And then what I've seen too is that some low concentration numbers may be more important than we see for other, aerobic pathways. When I see low numbers, because they're more ubiquitous, I don't, hold a lot of credibility for what's going to happen there. But when we're talking about anaerobic pathways like this, it makes me look at those low concentration numbers a little more closely. Do you feel the same? Yeah,
I think that makes sense. I mean, there are papers that have talked about how the critical players are minor members of the community for some of these targets, particularly with hydrocarbons. It's such a complex mixture, and when we look at a regulatory driver, it's not the only thing there. But important processes could be happening even with those low numbers. So I agree that makes sense.
And one final question as we end today is, we talked a lot in your presentation about anaerobic xylene, and one of the concerns always is benzene at sites. Have you done any research in anaerobic benzene biodegradation and kind of your thoughts on what we know on some of those genes?
Sure. Well, that's Elizabeth Edwards, one of her favorite topics and something she's been working on for a long time. And she's been doing meta-omics on benzene degrading cultures and certainly making progress. It's a tough nut to crack, and so we haven't really worked on benzene. I mean, we've certainly wanted to and given it some thoughts, but we've been working with Elizabeth. She shared her O-xylene culture, and I know she's doing similar things with the benzene culture, and of course, she does fantastic work. So I would keep my eyes on Elizabeth for benzene news.
Okay, great. Well, thank you very much, Susan, for your time today. We appreciate you diving into this. And again, if there's additional questions that you all have, you can email them to Susan. Her email address is there, or you can send them to info@microbe.com, and we'll be happy to answer those for you. But again, thank you, everyone, for your attendance today, and thank you, Susan, for taking time out of your busy schedule to do this webinar for us.
Sure. No, thank you, Dora, for the invitation. It was fun, and I hope some good conversations come out of it.
All right. Thank you, everyone.
All right. Bye.
Thanks. Bye-bye.